Did you know that viruses can be used to treat diseases? Although it might seem impossible for viruses that infect millions of people every year and cause so many different types of diseases to have any positive impact on humans, the reality is they can be used as a great vehicle to treat genetic diseases. Of course, you might be asking, but how can they be used to treat genetic diseases? To answer your question, we first need to explain what gene therapy is. Gene therapy is a technique that modifies a person's genes in order to treat or cure a disease. A gene is a sequence of nucleotides in DNA that can encode a protein. DNA is a biological polymer found in human cells, which is made of chemical building blocks called nucleotides. A nucleotide is made of 3 parts: a sugar group, a phosphate group, and one of 4 types of nitrogen bases. The chains of the DNA are attached to each other using the hydrogen bonds between each nitrogen base. The nitrogen bases are adenine, thymine, guanine, and cytosine. So basically, multiple nucleotides in a sequence create a gene. And genes can encode proteins that have certain functions in our bodies. If a gene that encodes a certain protein in our bodies was missing or mutated, then serious health problems can occur. Gene therapies can work by several mechanisms. Either by replacing a gene that causes a certain disease with a healthy copy of the gene, or inactivating a disease-causing gene that doesn't function properly, or introducing a modified or new gene into the body to help treat a disease. Gene therapy products are being investigated for their ability to treat diseases including cancer, genetic diseases, and infectious diseases. Gene therapy products can be created using different methods, including the use of plasmid DNA. A plasmid is a small circular DNA molecule that is found in bacteria and some other microscopic organisms. Plasmid DNA molecules can be genetically engineered to carry therapeutic genes into human cells. The use of viral vectors, since viruses have a natural ability to deliver genetic material into the nucleus of cells, and therefore can be modified to remove their ability to cause infectious diseases. These modified viruses can be used as vehicles to carry therapeutic genes into human cells. The use of bacterial vectors. Bacteria can also be modified to remove its ability to cause diseases, and then used as a vector to carry therapeutic genes into human cells. The use of patient-derived cellular gene therapy products, where cells are removed from the patient and genetically modified and then returned to the patient's body. When gene therapy is used to modify cells outside the body, blood, bone marrow, or another tissue, is taken from a patient, and then specific cell types are separated in the lab. Afterwards, a vector containing the desired gene is introduced into the separated cells. The cells are later injected into the patient, and the new gene is used to produce the desired effect. Currently, viruses are the most commonly used vectors in gene therapies. Before a specific gene therapy can be marketed for use in humans, the product must be tested in clinical studies for safety and effectiveness. Now, your next question might be, but how can we use gene therapy that utilize viral vectors to treat blindness? There are many different companies that have been working on gene therapies to treat diseases caused by genetic mutations, which cause blindness. In 2017, Luxturna, a gene therapy which was developed by the US company Spark Therapeutics, became the first gene therapy for inherited blindness to receive FDA approval. The therapy is designed to treat patients with mutations in a gene called RPE65, which encodes a retinal protein that is essential for the eye to respond to light. The treatment consists of injecting a healthy copy of the gene into the eye, using a viral vector, to restore the missing protein. The virus is modified to eliminate its capacity for infection, and the functional gene is introduced into its genome so that it can be used by the patient's cells to generate a functional protein. Only one injection in each eye has been shown to be enough to improve vision for at least 3 years. Gene therapy is really well suited to treat the eye, since the eye is an area of the body where the immune system has restricted access. This prevents immune reactions against the virus used to deliver the treatment. In addition, the cells of the retina can keep the injected DNA functioning for longer than other cells in the body. This means that a single dose of the product injected into each eye can restore sight in the long term. GenSight is another company that has been working on a gene therapy that utilizes a virus as a vector to treat blindness. The company developed a product that treats Leber hereditary optic neuropathy, a disease that causes irreversible vision loss. So far, the treatment has been shown to be safe and has been shown to maintain its effects for at least 5 years. GenSight is also developing a treatment that combines gene therapy with a wearable device. The gene therapy introduces a light-sensitive protein into the optic nerve cells. Then, a pair of goggles helps redirect and concentrate the light toward the cells, which will then convert it into a signal for the brain. This type of therapy could potentially treat all types of retinitis pigmentosa. Retinitis pigmentosa is a group of rare eye diseases that affect the retina. Another company that has been testing gene therapy for eye diseases is Biogen, a US-based pharma company that has been testing a gene therapy for choroideremia, a condition that impairs night vision. Thank you so much for watching the video, and we hope you found it informative. Please consider subscribing to our channel if you enjoyed the video.